Welcome to While We Were Distracted, the podcast where we explore topics that often get overshadowed in the rush of daily life. Please take a moment to like and subscribe, it really helps.
Over 1 million Americans lost. A global shockwave. We know you don't have 2 hours to watch C-SPAN, but understanding the COVID-19 story is crucial.
Absolutely. So today, we're doing a deep dive. We're taking that recent Senate Homeland Security Committee hearing on COVID origins.
And boiling it down, we'll pull out the key insights, the core arguments, basically everything you need to know about where things stand.
And it's vital, right? Because this isn't just about looking back. It's about future health security. Yours, mine, everyone's.
Exactly. So the big question hanging over that hearing and still hanging now is, how did this pandemic actually start?
Right. Two main ideas are still on the table.
Hmm.
You've got the natural spillover theory, jumping from animals to humans.
Or the other possibility, something related to a lab incident.
And look, the hearing didn't give us a final, definitive answer. That's still elusive. But the goal was to shed light on the scientific evidence.
Get it out in the open. Our mission here is similar: distill those 2 hours of testimony, the arguments, the evidence, so you get the picture quickly.
And understand the implications. What does this debate mean for preparing for, well, whatever might come next?
Okay, so let's start at the beginning. How did this Senate investigation kick off? What was the setup?
Well, the Senate committee, led by Chairman Peters, launched it to really examine the science and aim for transparency for the public.
And Senator Paul emphasized it was a bipartisan effort too, didn't he, this biosecurity investigation?
Yes, he did, highlighting that there's a shared interest across the aisle in figuring this out and boosting our preparedness for biological risks.
It's worth remembering something Dr. Gertz pointed out in the hearing: finding the origin of any outbreak. It's often really hard.
Oh, definitely. It can take years. Sometimes you just never find that patient zero moment or the exact source. COVID's proving to be, well, a tough case.
So the hearing dove deep into those two main theories, natural versus lab-related.
Exactly. And one of the really striking things that came out was about the initial private thoughts of some key scientists right at the very start.
Yeah, this was fascinating. Thanks to FOIA requests, we saw emails from people like Anderson, Lipkin, Gary, Barrick, and privately, they weren't dismissing the lab idea.
Not at all. Anderson, for instance, in one email, called a lab escape thing likely. Pretty strong words.
And Bob Gary, looking at the virus's genetic makeup, privately questioned if a natural jump could really explain those specific features he was seeing.
Right. He struggled, apparently, to see how those genetic insertions could have just happened naturally altogether like that.
Even Ralph Barrick, who's a major figure in coronavirus research and collaborated with the Wuhan lab, admitted privately the lab's huge virus collection and its location were, quote, a problem.
And then there was Dr. Fauci. His initial private emails also showed concern about gain-of-function research happening in Wuhan and mutations that looked possibly engineered.
Which contrasts, pretty sharply with some of the later public statements strongly downplaying the lab leak theory.
It does. And this leads to what Senator Paul called a remarkable reversal.
Which was,
Well, despite these private doubts flying around, some of those same scientists, Anderson, Lipkin, Gary, co-authored the proximal origin paper.
yes, the paper that basically concluded a lab origin was implausible.
Exactly. So that gap, that divergence between the private concerns and the public conclusion, that's become a real focal point in this whole debate.
Okay, so that sets the stage for the private doubts. What actual evidence was presented during the hearing that pointed towards a possible lab origin?
A key area was the timeline. There's mounting evidence suggesting the virus was actually spreading, perhaps in Wuhan and even wider, back in the fall of 2019.
So before the first official cases linked to the Huanan seafood market in December.
Correct. We heard about scientific modeling estimating an earlier emergence, reports of Wuhan hospitals being unusually busy that fall.
I remember hearing about antibodies found in blood samples from Europe and elsewhere, taken very early in 2020 or even late 2019.
Yes, and wastewater analysis finding traces. Plus mentions of illnesses at the Wuhan military games in October 19 and unexplained school closures in Wuhan around then, too.
Yeah.
It all paints a picture of potentially earlier spread.
Then there was the market data itself. That seemed confusing.
It did, because despite a massive amount of testing, they couldn't find a single infected animal. Not in the market, not in the supply chain feeding it.
Which is weird, right?
Yeah, it is. And all the early human cases identified were lineage B, which scientists think is likely not the original ancestral strain that jumped.
Okay. And the environmental samples. They found virus traces, but
not much, apparently. They found animal DNA, yes, but not significant levels of SARS-CoV-2 co-located with it. which again is quite different from SARS-1 back in '03.
How so?
With SARS-1, the market animals, like civets, were heavily infected. It was much clearer. Here, the animal link at the market is, well, much harder to pin down.
Senator Paul also mentioned something about raccoon dog data being misinterpreted.
Yeah, he called it shameful. Some initial reports hyped up finding virus RNA near raccoon dog DNA, but the reality seems to be the viral load was tiny and in very few samples. Not exactly a smoking gun.
Beyond the market, there were also questions raised about the Wuhan Institute of Virology itself, weren't there? Events happening in 2019.
Yes, starting around March 2019, things like unusual attention from the Chinese Communist Party, a military physician being put in charge of aspects of the lab.
And big spending on biosafety gear and security upgrades.
Exactly.
Mm.
Plus, reports that the lab's main virus database went offline, vanished. And this very concerning illegal shipment of dangerous viruses from Canada to Wuhan, one of those was later found contaminating samples in another lab.
So a lot of activity and some potential red flags around lab operations and safety.
It certainly raises questions. And then there's the flip side. The lack of strong evidence for a natural origin, meaning they've tested tens of thousands of animals across China and Southeast Asia. Still no confirmed intermediate host animal carrying a virus close enough to be the direct ancestor of SARS-CoV-2.
And the blood bank samples from Wuhan before the outbreak?
Didn't show the expected antibodies. You'd think if it was spreading naturally beforehand, some people would have been exposed and developed immunity. But they didn't find that evidence.
Dr. Quay talked about something called posterior diversity. What's that?
Ah, right. Think of it like this. When a virus jumps naturally and starts spreading, it usually develops lots of little genetic variations early on as it adapts. That's posterior diversity.
And SARS-CoV-2 didn't have much of that.
Apparently not, especially compared to what you might expect from a natural spillover. Its early genome was relatively uniform. He also noted how the outbreak seemed laser-focused on one market, Quanon, unlike SARS-1, which popped up in multiple markets.
Okay, perhaps the most technical and maybe controversial part was about the virus's genome itself, features suggesting it wasn't natural.
Yes, Dr. Quay laid out about 8 specific features he argued strongly suggested lab manipulation, or at least were incredibly unlikely, 1 in a billion probability, he claimed to have all arisen naturally.
What kind of features are we talking about?
Things like the underlying genetic backbone, the specific shape of the receptor binding domain, the part that latches onto our cells, and crucially, the furin cleavage site.
That's the bit that makes it more infectious for humans, right? And it's unusual.
Very unusual among this type of coronavirus, found in SARS-CoV-2, but not in hundreds of its closest known relatives. He also pointed to sequences that look like genetic stitching points, matching methods used in labs, like those pioneered by Dr. Barrick, and the ORF8 gene.
And he mentioned a grant proposal.
Yeah, 2018 DARPA grant proposals from EcoHealth Alliance and the Wuhan Institute. Quay claimed there was a 14 out of 14 match between the unique features of SARS-CoV-2 and the exact kind of manipulations described in that proposal, which aimed to create viruses with novel spike proteins and furin cleavage sites.
Wow. So his argument culminated in this idea that Wuhan had the motive, the means, the expertise.
Exactly, a thought experiment.
Yeah.
You have this global center for coronavirus research, doing gain-of-function work, synthetic biology, animal experiments, and even drafting blueprints for a virus with precisely these features. It's suggestive, to say the least.
Okay, but there were strong counterarguments defending the natural origin theory, too. We need to cover those.
Absolutely.
Yeah.
Dr. Gary, for example, really emphasized the geographical clustering of the very earliest known cases around the Quanon market. That points to it being the epicenter.
And he mentioned molecular evidence from the market?
Yes, specifically finding SARS-CoV-2 RNA physically mixed with genetic material from animals like raccoon dogs and civets, especially in that southwest corner where live wildlife was sold.
So virus and susceptible animals in the same place at the same time.
That's the argument. And genetically, he suggested the data points towards potentially 2 separate jumps from animals to humans happening in the market.
He also pushed back on the idea that bat viruses could jump directly, right? Saying an intermediate animal is usually needed.
Correct. That's been the standard thinking for coronaviruses. He also stated clearly that there's no actual proof the SARS-CoV-2 virus itself existed in the Wuhan lab before the pandemic began.
Though he did concede the lack of Chinese transparency makes that hard to verify definitively.
Yes, that lack of transparency was a constant theme. He also brought up another virus, Benel 2052, found in bats.
How is that relevant?
It's genetically quite similar to SARS-CoV-2 in key areas, including the receptor binding domain. His point was that features that look unusual or engineered can, in fact, arise naturally in the wild.
Okay, this inevitably leads us into the whole debate around gain-of-function research. Let's unpack that a bit.
Yeah, it's a crucial and often contentious part of the story. Simply put, it's research that aims to modify a pathogen, potentially making it, say, more transmissible or more virulent.
And proponents, like Dr. Gary at the hearing, argue it's necessary.
Yes, he argued it's vital for understanding how viruses might emerge or evolve, for developing animal models to test vaccines and treatments, basically for getting ahead of potential threats. He mentioned successes like flu research, the HPV vaccine, emphasizing it must be done responsibly with strong oversight.
But the critics, like Dr. Ebright and Dr. Quay, have a very different take.
A very different take. They argued that this kind of research on potential pandemic pathogens offers very little clear civilian benefit that couldn't be achieved through safer means, while posing, frankly, unacceptable biosecurity risks.
Dr. Ebright pointed back to concerns raised years ago, right, about work at the WIV.
Yes, back in 2015, concerns about experiments creating enhanced, more dangerous SARS-like viruses. And he drew that line again to the 2018 DARPA proposal, suggesting it outlined work very similar to what might have created SARS-CoV-2.
And the biosafety level at the Wuhan lab was questioned, too.
Heavily questioned. Dr. Ebright cited evidence suggesting much of the risky bad virus work was done at biosafety level 2, BSL-2, which is, well, far below the BSL-3 or BSL-4 generally considered appropriate. He reminded everyone that lab accidents happen. Even at top facilities, SARS-1 leaked multiple times. Even SARS-CoV-2 itself has leaked from labs since the pandemic began.
Dr. Gertz mentioned a slightly different angle, too, gain of opportunity.
Right. This highlights the risk that comes just from collecting thousands of novel viruses from the wild and concentrating them in one place, like a lab. Even without intentionally modifying them, just having them there increases the opportunity for accidental exposure, escape, or unexpected natural recombination.
Okay, beyond the science, the hearing really hammered on issues of oversight and accountability. That seemed like a major focus.
Huge focus. Firstly, the immense frustration with China's lack of transparency, not providing raw patient data, withholding research records, deflecting investigations. That hinders any origin investigation.
But there were also serious allegations aimed closer to home at U.S. institutions.
Yes, accusations of a potential cover-up or at least a lack of transparency from some U.S. government quarters. Things like withheld Fauci emails, the ODNI refusing to declassify relevant intelligence, and reports of key NIH officials, like Dr. Morans, deleting emails related to EcoHealth and the Origins investigation.
And specific failures cited regarding how NIH managed the grants funding the Wuhan research?
Definitely. The fact that EcoHealth Alliance, the intermediary, apparently failed for years to ensure the Wuhan Institute provided required progress reports and data, with little consequence from NIH. This led to calls for debarring EcoHealth WIV from future funding.
And holding NIH officials accountable for those lapses.
Exactly. There was debate on whether that accountability has truly happened.
Dr. Gertz also flagged a loophole in U.S. rules, didn't she, about privately funded research?
She did, pointing out that research with dangerous pathogens, if funded privately, currently falls outside federal oversight frameworks like the P3CO policy.
And that MIT Red Team experiment sounded pretty alarming.
It really did. It showed how easily a team, even following current biosecurity guidelines, could acquire the genetic materials needed to reconstruct dangerous pathogens, highlighting weaknesses in the current system.
So, given all these issues, the scientific uncertainty, the safety concerns, the oversight failures, what were the main recommendations coming out of the hearing for moving forward? What should we do?
Several key themes emerged. First, a strong need to beef up biosurveillance, both here and globally, to catch new threats earlier.
Makes sense. And the One Health idea.
Yes, integrating human, animal, and environmental health monitoring to better predict and prevent zoonotic spillover events before they become human pandemics.
There were calls for a stronger global framework, too, right, for managing biorisks internationally.
Definitely. And critically, several experts argued for an independent body within the U.S. to oversee high-risk biological research, separate from the agencies that actually fund the research, like NIH or DoD, to avoid conflicts of interest.
That seems like a big structural change.
It would be. The idea is to have oversight that covers all potentially risky research, no matter who funds it or whether it's classified, and, as Dr. Ebright stressed, make this oversight legally enforceable, not just guidelines.
Any other concrete proposals?
Yes. Things like tighter export controls on advanced biotech equipment that could be used for risky research. thinking more carefully about where high containment labs are located, considering those gain of opportunity risks associated with large virus collections.
And more transparency from our own intelligence agencies about what they know or suspect regarding the origins.
That too, a call for more declassification and sharing.
So wrapping this deep dive up, it's clear we're still grappling with huge questions. Conflicting science, serious allegations flying around, a fundamental debate about how we conduct research safely.
Absolutely. The stakes couldn't be higher, really. This whole experience underscores the potential for future pandemics, maybe even worse ones. Learning the right lessons now is just critical for preparedness.
Which leads us to a final thought, something for you, our listeners, to mull over. Even if we never get that 100% certain answer on how COVID-19 started.
Which we might not.
This hearing undeniably exposed major vulnerabilities in lab safety, in research oversight, in international cooperation, in government transparency.
So the question becomes, regardless of the origin, what actions do you think are the most essential right now to patch these vulnerabilities? What needs to change to safeguard our future against the next biological threat? It really touches on trust in science, holding institutions accountable, and ultimately global health security.
A lot to think about. Thanks for joining us on this deep dive.
Please take a moment to like and subscribe. It really helps.